Hello everyone. Today we are discussing about recombinant DNA technology. We are giving an outline for the recombinant DNA technology. What are the steps and what is this and what is the history and everything we will discuss in this video. So these are the contents that we are going to discuss. And you can see the recombinant DNA technology, the DNA molecules which are extracted from different sources and they are chemically joined together. That is known as recombinant DNA technology. Okay, so say if we tell that it is from a same source which is not a recombinant one. We extracted and chemically joined DNA from different sources, then only it is termed as recombinant DNA technology. Okay, and then these are the history of the recombinant DNA technology. As we know that the discovery of DNA structure which is proposed by Watson and Crick in 1953, and then the isolation of DNA which is firstly carried out in 1967, and then the restriction endonuclease which is isolated in 1970, and then generated rDNA technology by Paul Burke in 1972, and 73 Cohen and Boyer which is produced the first plasmid vector. So these are some milestones in the discovery of recombinant DNA technology. Okay, and what are the goals of recombinant DNA technology? First of all, it is nothing. It is to isolate and characterize a gene. And then after isolating and characterizing, we can make desired alterations in one or more isolated genes. Okay, then only we can produce more competent DNA segments. So that the first step is the isolation and characterization of a gene. After that, we can make desired alterations. And then the next step or the next goal to return this altered genes into a living cell. Once we take out the DNA from the living cells, after that we are making the alterations. After that, then we have to return this altered genes into a living cells. Okay, so that of course it means that we are artificially synthesizing the new genes, and this alternating or the alteration of the genome of an organism. And another goal is the understanding of hereditary diseases and their cure or their treatment, and of course the overall improvement of a human genome. These are the goals of recombinant DNA technology. And what are the procedures of making this recombinant DNA? How can we create a recombinant DNA? The first step is, of course, it is the isolation of DNA or taking out the DNA from a living organism, and then we have to cut the DNA. Then only we can make alterations to that particular DNA. And the third step, the cutting DNA have to join together. That is the third step. And after that, now it is making a recombinant DNA. After that, we have to amplifying this recombinant DNA further. Okay, these are the 4 steps that depicts the recombinant DNA technology. The first step, that is the isolation of DNA. So that how can we isolate the DNA? We know that the DNA which is extracted from the nucleus. Okay, so that we have to lyse the cells. So the first step is the cell lysis, which is using a detergent. So that this detergent which will disrupt the plasma membrane of the cell. After that, the cell contents are treated with the protease. So what is the function of the enzyme protease? Of course, it is the destruction of proteins. And another one is the RNase enzyme. So what is the function? In order to destroy the RNA. First of all, we lyse the cell wall and the plasma membrane. And after that, we are treating with the protease to destroy all the proteins in the cells and also the RNAs to destruct all the RNAs. Then we undergoing for a centrifugation process, all the cell debris which are pelleted in a centrifuge, and the supernatant liquid which containing the DNA, and all the cell debris which are sedimented. Okay, so that the supernatant which will transfer into another clean tube, and the DNA is precipitated with ethanol. It forms viscous strands that can spotted on a or spooled on a glass wood. So that is about the isolation of DNA. After isolating the DNA, the next step is the cutting of DNA. The DNA which can be cut into large fragments by mechanical shearing. After that, after cutting the large fragments, the next step is we are using the enzymes, restriction enzymes, which are actually cutting the specific size. There are different kinds of restriction enzymes are there, and this restriction enzymes or the restriction endonuclease, which are known as the molecular scissors. The enzyme restriction enzymes which are known to be the molecular scissors in genetics. So the restriction enzyme which is found in bacteria and which are site specific cleavage. Through the mechanical shearing, we can cleave the large DNA fragments, or we can make large DNA fragments. After that, we are using this restriction enzymes to make a site specific cleavage. And this restriction endonucleases which recognize DNA based sequence which are palindrome. So what do you mean by palindrome? You can see in this picture, there is a palindromic sequence which can read both forward and backward at the same time or the same manner. So that is known as the palindromic sequences. This restriction endonuclease which makes staggered cuts. What do you mean by staggered cuts? Which makes sticky ends, or they are having complementary sequences. So they can recognize complementary sequences so that they can tell that it is making staggered cuts or sticky ends. The third step is the joining of DNA. So of course, in the second step, we are cutting the DNA by using the restriction endonucleases. So that the next step is we have to join together the DNA fragments. So that is the third step, the joining of DNA. You can see the detailed steps here. The plasmid, you can see the cleavage site, and we are using the restriction endonucleases there which will produce the sticky ends. After that, we are inserting another foreign DNA, that is in blue color here, the foreign DNA. The same manner we are using restriction endonucleases, the same restriction endonucleases here, the EcoR1. So by using this EcoR1 endonucleases, we make cut into the plasmids and also the foreign DNA. After that, we are inserting the foreign DNA into the plasmid. Okay, then we have to join these two DNAs together so that we are using the enzyme DNA ligase. Okay, so since the restriction endonucleases are known as the molecular scissors, the same time the DNA ligases are known as the molecular glue, or we have to paste together by using this DNA ligases. After that, we can get the plasmid chimera. What do you mean by chimera? Which is having the DNAs from two different sources, so that which is termed as plasmid chimera or recombinant DNA. The third step is the amplifying the recombinant DNA. What do you mean by amplifying? which means we are enhancing the recombinant DNA numbers. For that, we are transforming the so-called plasmid chimera into a bacterial host strain. Why we are using this bacterial host strain? Because bacteria are having very short life cycle. They can multiply in large numbers in very short time. So that once we inserted our plasmid chimera or the recombinant DNA into a bacteria which will multiply in very short time, so that we can get large numbers of recombinant DNA. For that, the cells are treated with calcium chloride, so that the cells become competent, because every cells or all cells are not competent to take the foreign DNA. In order to enhance their capacity of transformation, we are treating the cells with calcium chloride. So the medium having the cells along with the calcium chloride, after that we are adding the DNA. And then the cells are heat soaked at 42 degrees Celsius. That will also enhance the capacity of transformation. The DNA which goes into the cell or that process is termed as the transformation. Once in a cell, the recombinant DNA will be replicated further. Okay, that is a natural process. When the bacteria undergoing division, the same time all the recombinant DNA that will enter into the cell by transformation will again replicated or will increase in their numbers. Okay, thus the DNA, the recombinant DNA is amplified. Okay, so you can see the steps in this picture, the target size and the recombinant DNA creation or formation, after that which will enter into the bacteria, that is known as the transformation, and then the replication of bacteria, and which will produce many number as a result of cell division. So what are the major enzymes used in recombinant DNA technology? Of course, the first one is the DNA ligase. I told you it is the molecular glue, which is used to paste the foreign DNA into the vectors. I mean the plasmids or somewhat like that. Okay, so which is using to bind the DNA molecules. And the second is the type 2 restriction endonuclease. So what is the speciality or what is the function? Which cleaves the DNA at specific sites. And the third important enzyme is reverse transcriptase. So what is the function of reverse transcriptase? As a result of normal transcription, DNA is converted into RNA. In the case of reverse transcriptase, the RNA which forming DNA, the reverse process. Okay. And the another enzyme is the DNA polymerase 1. So this DNA polymerase 1 which will fill the single stranded gaps of DNA duplex. Sometimes the process is not that much accurate. They may have the errors. Okay, sometimes which may produce some gaps in the DNA duplex. So this DNA polymerase 1 which will fill such kind of gaps in the DNA duplex. And another one is polynucleotide kinase which will add the phosphate group to 5' OH end of a polynucleotide. And another one is terminal transferase which adds homopolymer tails to the 3' OH ends. And then another enzyme is exonuclease 3. So what is the function? Which will remove the nucleotide residues from 3' end, or which will removes the nucleotides from one end, that is known as outside, that is known as the exonuclease. Whereas in the case of endonuclease, which will make or which will make a cut within the within the strand, that is known as the endonuclease. Exonuclease means which will remove the nucleotides from one end, that is from 3' end. Okay. And another one is the bacteriophage or lambda exonuclease, which removes nucleotides from 5' end. Exonuclease 3, which will remove nucleotide residues from 3' end, whereas the bacteriophage exonuclease, which removes nucleotides from 5' end. And another important enzymes which is used in recombinant technology is the alkaline phosphatase, which removes, phosphatase means removal of phosphate group. Okay, which removes the terminal phosphate group. And then we have to discuss what are the vectors used in rDNA technology. So, what is a vector? Which means it is a vehicle. Vector means it is nothing, it is a vehicle. So, once we made the recombinant DNA, then we have to insert into the living cell through one agent called a vector. Okay. So vector, what are the functions or what are the characteristics? A vector, which is an area of DNA that can join another DNA or the foreign DNA part without losing its limits of cell replication. Okay, once it loses the self-replication capability, there is no meaning for using this recombinant DNA technology. So that exclusively they are having the capacity for self-replication. And they should be capable of replicating in host cell also. Another characteristic that should be capable of replicating in host cell. And then they are having convenient restriction endonuclease size, then only it will make the suitable cuts for inserting DNA of interest. If they don't have the restriction endonuclease site, we never make a cut in the plasmids or somewhat the area. And they should have a selectable marker to indicate which host cells are received recombinant DNA molecule. It is known as the selection process because we don't know whether all the cells they are transforming or non-transforming cells. So that in order to select the transformants from the non-transformants, we are using certain selectable markers. Okay, usually we are using the antibiotic resistance genes as the selectable marker. And then another is character that should have for a vector is there should be small and easy to isolate, or they may be in small size. These are the vectors which are used in rDNA technology. You can see that the BAX means the bacterial artificial chromosomes, YAX means yeast artificial chromosome, plasmids, lambda phage, plasmids, expression vectors, and binary vectors are also there. So these are the vectors which are used in rDNA technology. The first one is the plasmid vector. The plasmids, which are small circular extra chromosomal DNA. Okay, they replicate independently of bacterial chromosomes, which means within the bacteria there is a bacterial chromosome. Apart from the bacterial chromosomes, these are the plasmid vectors or plasmids. These plasmids, they have a ability to self-replicate without depending on the bacterial chromosome. These are useful for cloning DNA inserts less than 20 kb. And the inserts larger than 20 kb are lost easily in the bacterial cells. So we can insert only small pieces of DNA, such kind of 20 kb. So that is about the plasmid vectors. And another one is lambda phage vector. So lambda phage vectors, which are recombinant, and what is the speciality of this bacteriophage? They can attack the bacteria easily, and they are transferring their DNA into the bacteria. So this phage chromosomes, in addition to embedded outside DNA. All in all, the phage vectors which can convey bigger DNA groups than the plasmid vectors. We see that the plasmid vectors, they can capable of transferring DNA of less than 20 kb. If we have the bigger ones, we are depending on the lambda phage vectors. And another one is the cosmid vectors. The cosmids, as the name indicates, the cosmids which are hybrids of the phage vectors and the plasmids, and which is also capable of carrying larger DNA fragments up to 45 kb, which are nothing, they are plasmids, but they are having a co-site of the phage vector. Okay, we will discuss these vectors in details later. Even though you just remember that these cosmids are which are hybrids of plasmids and the phage, and in other terms it is termed as the plasmids, they are having co-sites, they are known as the cosmids. They can replicate like the plasmids but can package it like the phage lambda. We will discuss the details in later. And expression vectors. So another kind of vectors are known as the expression vectors. What are expression vectors? The so-called, the so detailed the plasmids or bacteriophage, they are just using for the multiplication. They cannot express their own genes. In that case, the expression vectors means they can carry the host signals and that facilitate transcription and translation of an inserted gene. The so-called the cosmids, whether it is a cosmid or it is a plasmid or it is a phage, in all these cases they cannot express the inserted gene. Whereas in the case of expression vectors, these vectors are capable of expressing the inserted gene. These are very useful for expressing the eukaryotic genes in bacteria. Okay, so that is the expression vector. We will discuss later in detail. And another one is one of the expression vector is yeast artificial chromosomes. Yeast artificial chromosomes or YACs, which are yeast vectors, and they have engineered to contain a centromere, telomere, origin of replication, and a selectable marker, which indicates that they are capable of expressing the foreign gene in themselves. So that it is a expression vector. I hope you got the point. Because these chromosomes, they are known as the artificial chromosomes, which means they are engineered to get a centromere, telomere, origin of replication, and selectable marker, since these are helping for them to express in themselves, so that the engineered or the foreign DNA can express in themselves, and they can carry up to larger fragments, which means 1000 kb of DNA, whereas in the case of plasmids, which is carry only 20, less than 20 kb, whereas in the case of cosmids, they can carry up to 45 kb only. When compared to that, these YACs are so far better, they can carry up to 1000 kb of DNA, and they are very useful for cloning eukaryotic genes which contain introns. Because compared to that of prokaryotic genes, eukaryotic genes are larger and they are having the introns also, so that they are larger in size. So in order to express these eukaryotic genes, we are relying on this artificial chromosomes. And bacterial artificial chromosomes, they are known as the BACs. These are bacterial plasmids derived from the F plasmid of the bacteria, and they can capable of carrying up to 300 kb of DNA, which is also having this origin of replication, selectable marker, and so on. You can see in this picture, so that they can express themselves. And another category of the vectors are the shuttle vectors. So shuttle vector, as the name indicates, somewhat they are shuttling between two organisms. It can replicate in at least two different host species, most probably which is shuttling between a prokaryote and eukaryote, so that the DNA recombined in that vector can be tested and manipulated in several cell types, whether it is there is no matter whether it is a prokaryote or eukaryote. One of the example is the yeast artificial chromosomes. They can be amplified in bacteria and manipulation of large DNA inserts in the yeast or yeast the Saccharomyces cerevisiae. And another example is the agrobacterium mediated or tDNA. They also shuttling between prokaryotes and eukaryotes. So these are about the outline of vectors that are used in recombinant DNA technology. And as we know that the RDNA technology, they are having vast applications in agriculture. For the most familiar, the GM foods are there and pesticide resistant fruits, and there are so many applications in the case of agriculture and pharmacology. As we know that the artificial insulin productions, drug delivery, and there are so many applications, and medicine also, gene therapy, antiviral therapy, vaccination, and other uses are also there, that is glowing plants, fluorescent fishes, and these are we'll discuss in another video. So that is about an outline or an introduction to the recombinant DNA technology. Thank you.